Sadly, for those of us talking today who've been involved in developing therapeutics in outbreak scenarios, when we talk about medical countermeasure product development, we consider two vastly different scenarios. We've got the emergency response situation, which we're in today, and then we have the peacetime or the preparedness phase to development. Now, Director General Tedros at the WHO meeting in Geneva in February said the world tends to cycle between these scenarios, but really in panic and apathy in our approach to treating or emerging infectious diseases. And it's our hope that what we do now in response to the COVID-19 does lead to an ongoing global effort in therapeutics preparedness so that we don't actually have to repeat this exercise in a few years' time. Today, I'll share some lessons learned from therapeutics development in both settings related to influenza, which will provide some context for Steve to talk about our approach in the COVID Therapeutics Accelerator. I'll present published data, and I must recognise the efforts from significant multi-sectoral collaborations involving industry, academia, clinical investigators, and of course the patients and their families underpinning all of the information, and I'm truly standing on the shoulders of giants. Now, the COVID situation is all too familiar. If you wind the clock back to pandemic H1N1 and H5N1, we were experiencing exactly the same challenges. Before the availability of a vaccine, we had a thin antiviral armamentarium at the time. Frontline clinicians, public health officials, and policymakers were facing an impossible situation: an emerging unknown virus, antiviral choice, dose, regimen, procurement, deployment. And the big question that we were wrestling with then, as it is today, is what is the drug? And importantly, what is the optimal dose? For every milligram above optimal is less for others, and manufacturing capacity is wasted. And for every milligram below optimal is giving the virus a chance to outsmart our interventions. Now, given the broad audience in today's webinar, it's really important for me to stress that any time we're talking about the science behind dosing, which is really a large focus today, we're talking about clinical pharmacology. It includes pharmacokinetics or PK, which is what the body does to the drug, and pharmacodynamics or PD, what the drug does to the body. Now, for efficacy, we're talking about an adequate time course of concentrations of the drug and at the site of the infection. For flu and COVID, this is the lung. For safety, it could be the impact of systemic concentrations or local concentrations on GI tolerability or cardiac ion conductivity and so on. And we're also talking about the many factors that can change dosing requirements by adding variability, by impacting both the PK and the PD. Factors like age, differing dosing requirements for the elderly, children or babies, or based on body size. The impact of comorbid conditions like renal impairment, hepatic impairment, or the significant anomalies that happen in critically ill patients. The impact of the disease course itself. Like COVID, we're focused on prophylaxis or post-exposure prophylaxis or early treatment, or moving through the spectrum to moderate and severe infection. Now, the viral kinetics and the transition to acute respiratory distress syndrome and systemic inflammatory response syndrome is very different at different stages of the disease. The impact of genetic polymorphisms may be more prevalent in different ethnic populations. The impact of food, diet, herbs, OTC supplements, or other prescription medications. All of these factors impact on PK and PD and can therefore mean differing dose requirements for an effective and safe regimen. So, when we're combining the science of dosing with all of these significant unknowns, we're trying to bring all of this together. It's really like weather forecasting with global consequences. So let's take a look at some of the lessons which we've learned from this experience. So the 5 lessons we've learned that we're applying our thinking to the COVID-19 Therapeutics Accelerator. We have to know what we're up against. It's really important. It's also sobering to look at the reality. We have to begin with multiple audiences in mind. And in the emergency response situation, you need a roadmap. You must have a highly integrated and agile team and the right culture. You cannot do checkbox drug development. You have to be smart. You have to be creative. And you can't do this alone. It requires a coalition of the competent. So the first lesson, this is really to know what you're up against. Developing therapeutics in a preparedness or a peacetime mode is hard enough, but doing it during a pandemic is formidable. Investigational or repurposed candidates bring their own unique challenges, and each compound must be evaluated individually. Of course, an investigational therapeutic may seem further away from the clinic at first, but just because a drug is approved does not mean it is fit for purpose. Is it really logical to think that a dose regimen that's been developed in HIV or malaria or rheumatoid arthritis will be fit for purpose for an acute virus infection in the lungs where in vitro susceptibility suggests a 10 or 100-fold lower potency? So as drug developers, we ask critical questions when triaging compounds, such as, what's our target confidence? Is this pharmacologically or biologically plausible? The ADME profile, as I mentioned before, what's the route of administration? How might organ impairment impact? What about age, ethnicity, pregnancy? Will the drug reach the site of infection at the rate and the extent to be impactful? Are loading doses necessary? And ultimately, what is the likely clinical therapeutic index and how might it be used in the real world situation safely? And during a pandemic, we have these other challenges to decision making. We have to balance the weight of emerging evidence. The clinical data is complex, it's often blunt and temporally challenging to deal with. Preclinical data is often very difficult to extrapolate. Yet we have these other key factors, the impact on procurement and deployment, stockpiling, manufacture, who do we deploy to, when, how, and the impact of those strategies on the epidemiology, and then trade-offs versus all of the other interventions that we could be considering. So the first lesson really is to know what we're up against. It's essential to recognise that the therapeutic intervention goals for an individual patient can diverge significantly from public health goals and differ to that from procurers. A treating physician wants to improve the clinical course of a patient and exposed individuals. A public health official is more interested in minimising viral shedding to reduce spread. And a procurer may be focused on where to invest the healthcare dollars in order to maximise impact of the entire healthcare system. So you must recognise this, and you must be prepared to be conversant in multiple lexicons instantaneously, as all these factors need to be considered depending upon whose shoes you're actually walking in. So to give you an example of an approach we've applied, where we've achieved the interdisciplinary linkage of PK/PD, epidemiological and health economic models into a single quantitative framework. This is deliberately geared towards candidate therapeutics where PK/PD information may exist, but the clinical evidence is sparse or lagging, like what occurs in a pandemic. Now, this published example shows a proof of concept where we explored pandemic planning scenarios for Oseltamivir against a range of potential virus strains, differing by infectivity and virulence. A simple SCIR model was done in the original paper, but this can be expanded to more sophisticated agent-based approaches in videos here, and they're quite useful to visualize. You can introduce 2 doses of an experimental therapeutic and incorporate its PK/PD variability. You can then determine the total number of infections across the course of an influenza epidemic, and this can be done at a meta-population level, which is the top panel, or a community level, which is the bottom panel. Those total numbers of infections at the end of an epidemic simulation can then flow into health economic tree-based models and provide a decision support framework for procurers and payers across a range of scenarios based on viral transmissibility and healthcare utilisation based on virulence. Truly bridging early pharmacology for experimental therapeutics all the way to the payer. But the take-home message here is less about the specific methods that have been applied, but rather if you want to accelerate candidate therapeutics during pandemic circumstances, you must be a polyglot. It's essential that you walk in the shoes of various stakeholders and be able to be conversant in several languages at once, as clinicians, individual patients, regulators and public health goals, and that of procurers and payers will diverge. So the second lesson is begin with multiple audiences in mind. Now, the next lesson is more about the ingredients for executing projects in high uncertainty environments. Now, once a clear objective is stated, like to credibly determine the optimal dose for a novel virus under pandemic constraints, the next thing that must be done is an initial roadmap is put down. Firstly, accept that the roadmap's wrong, but a destination and a roadmap gives a team a sense of purpose and directionality. It helps them plan for contingencies. This was actually the initial roadmap that was done for Oseltamivir dose optimization for novel influenza strains. Actually stood the test of time pretty well and has been presented quite broadly. As you can see, it's a complex and multifaceted problem. Requires input from virology, biomarkers, clinical pharmacology, CMC. So it's essential that you have a cross-functional and highly integrated team, and they will continue to shape this roadmap. Now, the competency of an integrated team is very important, but if you don't have the right team dynamics and mindset, you cannot possibly execute drug development activities in pandemic setting. The team has to be very prepared for a very different journey of drug development. In particular, ambiguity and the rate of change. Every day, new data challenges your hypotheses, thinking, experimental design from the day before. This means that one is focused on progress, not perfection, although scientific credibility remains core. It's much more aligned to crisis management. You have many small initiatives. You move forward in rapid loops. It's Darwinism at its core. And one has to celebrate the failures as they actually move you towards the successes. So the third lesson, really, it's relevant to our approach in the accelerator, is that you have to have a strategy. You've got to have an integrated team, an agile implementation, but you also have to have the right culture. Now, the weakest link in developing therapeutics during a pandemic is dogma. We've always done drug development this way. The traditional drug development cycle measures time in years. Our need for therapeutics for COVID is measured against a very different time scale. It's the nightly news reports of infections and deaths that we're hearing. So if we apply drug development principles like we have always done and expect that we can accelerate the access to COVID-19 therapeutics in a useful time, we are by definition insane. So the next lesson is that we have to be smarter in how we approach drug development. It's essential that we apply advanced data science and contemporary drug development methods, including model-informed drug development. Not to cut corners, not to avoid critical and objective decision-making, but to increase parallel processing and make the most of the available information content to cut time and accelerate the development and access to critical medicines. So firstly, the randomized controlled trial is considered the gold standard in evidence gathering. But in rapidly evolving situations like a pandemic, there can be significant changes in emerging information, loss of placebo arms due to policy changes, pressure of social media disrupting the scientific process, amendments, heterogeneity, as well as logistical challenges as executing these large studies. It can make definitive conclusions from RCTs challenging, and the timely availability even more so. Federated approaches like the WHO master protocols are one important way to drive harmonisation of efforts, particularly around aligning information that's collected and also endpoints. But there are also other ways to think about this, on creating drug development platforms with other types of data and making decisions on a totality of evidence. Small-scale clinical trials where PK/PD is explored can be powerful in teasing out drug effect and support the determining of optimal dose. We can improve the quality of emergency use or compassionate use programs to gather higher quality data, including PK/PD. Now, the integration of clinical pharmacology thinking into trials is not at all obvious for some. In March, when we finalised this commentary for CTS of more than 20 trials on clinicaltrials.gov, it wasn't obvious that any of them really appeared to examine dose ranging, PK or exposure response. And this would mean the trials designed themselves would lack the capacity to offer optimal dosing guidance to support decisions like optimal manufacture, procurement and deployment. Now, in the same way you don't prototype 50 jumbo jets, you use computer simulations to test the engine, the airflow dynamics, refine, adapt, learn, and modularize to assemble one jumbo jet for a test flight, we must fully apply biosimulation to our clinical development thinking. The application of math to pharmacology and biology is not new. Neither is its place in the drug development process, but it has never been more important than now with COVID. And we must deploy biosimulation routinely and strategically to provide efficiencies in overarching development programs and clinical trial designs for adaptability and speed, reduced costs, as well as improved decision-making in the face of this tsunami of grey data, which we're actually experiencing. And finally, using historic and related real-world evidence data and available quantitative methodologies to support decision-making is very important. The use of real-world registry studies can be used to tease out regimen effectiveness via targeted deployment of registries, regions, and sites. So the final lesson is that we must do this together. Everyone in the life science community has a role to play in accelerating the development of therapeutics for COVID. Even in emergency times, it's essential, though, for us all to be disciplined and apply critical thinking, scientific method, and rigour to our work. Now, there's a strong desire to do something that sometimes overrides the ability to contribute something impactful. And sometimes the effort to do something or publish something can misdirect attention and also resources. Now is a time for us all to play to our strengths: industry developers, clinicians, scientists, public health practitioners. This will ensure that we've got the best efforts placed in the right places so we can get the right treatment at the right regimen in the right patient at the right time. It's also important at this moment to support hyper-transparency and data sharing in alignment with fair guiding principles. The future depends on what we do in the present. Let our COVID legacy be improved global preparedness for the next disease X.